Hi, my name is Chris Mancuso, and today I'll be talking a little bit about a software package called GenePlexus, which is a web server and a Python package to do gene discovery using network-based machine learning. So what is the problem that we're trying to solve here is if someone has a gene set of interest, can we predict other genes that are relevant to that set? So how do people get gene sets of interest? So there's a lot of databases out there that cover diseases, processes, traits, gene ontology, Mondo, KEGG, and just to name a few. People can also get these from individual experiments. Some people can do case control experiments that are unique to whatever context people want to study, and then they can get a list of differentially expressed genes. However, oftentimes these sets are either incomplete or they could be noisy, containing false positives. So what we want to do is leverage the power of genome-wide molecular networks to kind of help refine the genes in this set and also be able to predict other genes that may be associated with that set. Networks are really great. They can vary in the different ways that they're constructed. So some networks just contain physical interactions. Some of those are BioGrid and the consensus path DB. Some networks are, they can integrate data from all kinds of different places. They can use physical interactions, they can text mine data, they can use information in databases, homology, and so you can build networks. This way, String is a very popular network that does this. So then you can also have networks that integrate data from thousands of gene expression data sets, and networks that do that are part of the giant networks. So also networks can be context specific. So they can be tissue specific. So the giant networks, you can have a different network for heart, different network for your lung, different network for your kidneys. Species specific, so BioGrid, String, these networks you can have be specific to a given species. And then they also be disease specific, where the connections maybe more relevant if you're looking at diseases. So we built a web server based kind of off of a method that we extensively benchmark. And the method basically is if you can represent a network as an adjacency matrix. So where the rows are genes and the columns are genes. And basically a square in the matrix is the weight between the gene M and gene N. And so what we want to do is we're just going to build a simple linear logistic regression classifier. And so what you can do is just take the rows. So if you want to look at a given gene, you can just use the connections of it to each of the other genes in the network. And you can label some of those genes as positives. And then we can also label some of these genes negatives. So for instance, we have here, I don't know if this is showing up good, but so what we can have here is these blue nodes are all positive nodes, and we can set a few, most genes as unknown, and then we can pick a handful of negatives, and then we can train a classifier that will return the probability of every gene in the network of how associated it is with the given input set. And so you can just use the same underlying network representation. And if you want to look at a different set, you simply just change which rows of this matrix that you're going to consider positive, negatives, and neutrals. So we've extensively benchmarked this. So we looked at a whole bunch of different processes, pathways, diseases. We used a whole bunch of different networks, a bunch of different validation schemes. And basically we benchmarked it against a method called label propagation, which is kind of the go-to method for network-based gene classification. And we pretty much show that this supervised learning method is usually always outperforms label propagation. And another thing that we did a little bit is that we tried comparing it to like graph convolutional neural networks and show that this supervised learning method actually works a little bit better than that. It's because those methods are a lot of data hungry. And so this problem, you only have a handful of labels. This method is, it's been shown to work at this. If you ever need to know anything about how these methods work or anything about networks and how function prediction, you can talk to Remy Liu in the group. He is he's an excellent person. So we're gonna a little bit about the software. So we're just gonna walk through this for a little bit, but before we do that, just answering a few questions. So which type of research will benefit? So we think that anyone who can generate a gene set of interest and then kind of want to get some more information about that gene set. So we're trying to design it to be sort of task agnostic, or you can pick the network that you want that might be better for you, but we really are trying to make this very general for anyone to use. What type of researcher will benefit? Hopefully everyone, hopefully these web servers and will help anyone, regardless of programming or network knowledge. Really, they can still use this method. Can it really be used to aid research? We think it can. We really extensively benchmarked this, so we think it kind of really can help people's research. Is the model context specific? And so the answer to that is yes. So you can, especially in the Python package, you can upload whatever network you want. So these networks are really what can make the predictions kind of much better for a given context. Also, is the model trustworthy? We try to do this. We do cross-validation, and we supply those results to the user so they can actually look when they get their results back. Did the model actually work on the gene set that I put in? And the next thing is the model and results understandable. We try to do this by providing some network visualization of the top predictions. Then also we compare the trained model to thousands of other pre-trained models, and we'll talk a little bit about that at the end of the talk. So what is the difference between the web server and the Python package? So the web server is accessed via the browser, so you don't need any programming experience. The results are all searchable and sortable on tables through web browsers. The visualization is done in D3, and it's all run on the cloud, so you don't really have to worry about any kind of hardware. So the Python package you can run locally, but you can also then easily scale it to running it the hundreds of times if you have compute clusters. But you can also just run it on the command line interface. And so basically, you can just install a Python package. There's like one line of code that you have to write on a command line, and then you can get all the results back. Okay, so stepping through the software. So the first thing a user is going to do is upload a set of genes that they care about. So they can either upload a file, they can copy and paste it. And so we take in a handful of different files, formats, and then at some point they're all converted to Entrez IDs because that's where all our models are, all our networks and all our gene sets, they all live in that kind of gene ID space. So they're converted and the user can see, the different attributes of the network, how many genes that they supplied are actually in that network. And which genes, how they were converted, what we converted them to. And then the next thing, which is the most complicated part for a user to understand, is which of these models, which of these parameters to choose. So you have to choose your network, and these networks vary in size, coverage, and construction. You have to choose how the network is represented in the machine learning algorithm. And so also how negative genes are selected. So we're not going to get into the depth of that right now, but on the web server, we've tried to provide guidance about when you upload something, which of these parameters you should choose. Also, I'd like to highlight that the pre-processing of all these networks and data sets and all these things is like the actual power of the web server. Running just a simple machine learning model is actually, it takes not that much time, but like having all of this stuff pre-processed beforehand is kind of what the benefit of the web server is. And then in the Python package, you can supply kind of your own gene sets and your own networks if that you think that will be more beneficial to your research. So training the model on the cloud. So this is for the web server. So the first thing a user is going to do is they're going to come up to the browser and then they're going to interact with the browser and then they're going to upload their set of genes and then they're going to also pick which network and stuff that they want to run it on. Then what happens is a resource is spun up on the cloud. So it's like a high resource computational node. So the web browser uses like a low cost thing. So it's just constantly on. Then as the job is submitted, a high resource, because these models sometimes take multiple 20 gigabytes to train. And so a resource is spun up. That resource then pulls in data from storage that is living on the cloud as well. Then a model is trained, a machine learning model is trained specifically given the input of the user. But we want to highlight that a model is being trained, a machine learning model is being trained given the user's input. So a lot of times what people do is they train a model, and what they do is when a user accesses it, they just do inference from the model. They just use the trained model and get some predictions. So what we're doing here is supplying a user, the machine learning is trained on the user set. So then what happens is the data is then written to storage, and that's all sent back to the web app, and then you can view the results via the browser. But so this is very scalable. So in a lot of web servers, what happens if you have a traditional web server, you have to have all of these components. So if you want to run a machine learning model that takes 20 gigabytes, if 4 people hit that, then you need 100 gigabytes of memory. And so every time someone hits that, you kind of have to do it, and then people then have to be queued, and you have to wait. On the cloud, you can really just spin up, hundreds of things at once. We have to pay for it a little bit, but all of these things are very cheap. It's like 5 cents a job. But it's, so we can it's really scalable to have as many people want to hit this as possible. And it was a big team effort between biomedical researchers and developers and cloud architects. So the Python package, you don't use the interface, you use the command line interface. You can have a job manager like Slurm, and then go to a compute cluster, and then you use local storage. So it's just a little bit different, but like the same basic architecture holds between what the Python package can do and what is done on the cloud. So what are some of the things the web server will return to a user? So the first thing is the predictions of every gene in the network, how associated that gene is to the user's applied input set. So we can see here in this thing here, what you're going to see mostly is that known genes are going to be very high, right? So you are saying these are positive, so they're going to basically show up as positives too. But then you can see down here in row number 9, there's a gene that was not in the positives that is very highly associated with this input set. And you can see, we can label it as novel, so it wasn't used during training. And so these tables are all searchable and sortable, so you can just type in novel and just get like the top novel hits that you want. Yeah, and then there's links out to the gene information, and so it's a little bit more information there. Some interpretability. So what we did is we kind of took the top ranked genes and then used the underlying network that the model was trained for, and then we show the connectivity of those top ranked genes. And so there's interactive sliders for the edge weights, the nodes counts, the mean gene probability. And then it's also fairly informative. You hover over a gene, you get the ID, and you can get a little bit more information about the gene that was in that model. So the other way that we try to do interpretability is similarity to other pre-trained models. So the model is a linear model, so that means every coefficient, which is a connection to every feature is a gene, and every gene is basically going to get a coefficient. So there's just a set of coefficients that you get. So what we've done is for every single network feature negative selection combination, we've pre-trained models on thousands of gene sets of known processes and known diseases. So what we can do is we can take the user's model and compare the weights of that model to weights of a whole bunch of pre-trained models. And so then we can kind of find out which model was most similar to the model that was trained here. And so we can do this, so it's a little bit like gene set enrichment. It is not gene set enrichment. Mostly what people do is you just take the top ranked predicted genes and they put it in gene set enrichment. Having talked to some people here, we'll probably actually implement that because people actually really want to see this. But this is a way of more interpreting the model that was trained. So some future improvements, we kind of really want to streamline the GitHub repo and possibly make a package just for the web server part so you can get all the web server things, after you run your own results, you can access them. We want to think about user accounts so people can save gene sets so other people can see gene sets that were really important or other people's results, and then possibly aggregate predictions across multiple models. And then also incorporating novels, other species. Right now it's just humans, so we can either, a couple of ways of doing this is a user supplies mouse genes, we convert them to human genes, and then we use human networks. Also, we would like to just include, other model species networks. I've also been working on models where we take human and other species models and then combine them together and then run them through this algorithm. And we've shown that it's kind of actually really helps prediction in any kind of species that we're looking at. Okay, so summary is, we just given a gene set of interest, can we learn more about other genes and then kind of in an interpretable way? So it's all open source, and hopefully it's well documented. So interestingly, like who will use this software? According to a talk I saw earlier by Pedrag, is no one will be using this because experimental biologists don't use any modern software, right? So they don't use anything, and we want to change this. Like we really are open to like any feedback that people have that will get this in the hands of people who can actually use these models to help discover things about, health and biology in general. So I'm part of Arjun Krishnan's lab. So he has got a lot of different people working here. So Remy is doing a lot of graph convolutional neural networks, or just graph-based predictions. Sarah, I just gave a poster on topology. Hao is doing some cross-species stuff. Stephanie is doing network-based stuff. We were trying to find endophenotypes of diseases. I could talk about a bunch of more. Kayla's doing age and sex specific stuff. So I'm not going to read everyone's things, but there's a lot of great work that Arjun is doing, and he asked me to do this. So if you would like to join his lab, he is eager to have you, especially if you're really into natural language processing or you really want to get into single cell genomics. That's where he really kind of wants to expand the lab. So thank you very much. This was a team effort from everyone in Arjun's lab, and then also these people who were in the data, I think it's called data analytics and management, the IT-ish people at MSU who did the web development and the cloud architecture for us. They're excellent people. So thank you very much.
Thanks so much. All right, let's take some questions for Chris. We can start here.
That was really cool. I hate to tell you this, but Peja also said that bioontologists don't talk to people who develop computational tools. Yeah. So yep. Hopefully you can change that. My question. So we produce gene sets. We look at enrichment of networks in these gene sets. Say I were to use the Plexus software, plug in my set of genes, what do I get out of it?
What is like the main, so like the main thing that we want to do is return other genes that may be interesting of that set as well. So if you know a handful of positives, you can look at other genes that might be highly related to that set. So that might be relevant to the set in whatever context that you kind of want to think about it. So it's diverse in that respect. And so some things, I was talking to some person, so if you have a lot of genes, right, and then you want to see about like how they're connected and you want to use a certain network to look at it, you could put them all in as positives, but the machine learning model will actually downweight things that are not really connected that well in the network. So you can have something that is a positive, but then when you do the prediction, it will have a very low predicted probability of being in the input set. So you might just disregard looking at that gene in the future. So those are just two kind of things that you might be able to get out of the software. Okay.
Okay, we have time for more questions. Does anyone else in the audience have a question? Or are there any questions in the chat? Okay, how does the method differ from similar methods such as Gene Mania?
Yep, so there are methods out there. So we extensively kind of benchmarked these based on against classes of method called label propagation. So what label propagation does, in a general sense, I know there's a lot of different flavors of label propagation, but a very basic one is just guilt by association. So you can just look at a network, look at a node, look at its neighbors. What is its neighbors labeled as? And if that has a high enough count, then that unknown node can get that label as well. And then there's different ways people have kind of implemented label propagation. So you can take network edges and diffuse them in a diffusion network and then kind of do label propagation. So we compared our method to label propagation, which is what, as far as I kind of understand, is what Gene Mania is considered. It's a class of label propagation. So we compared our method versus that, and we basically always show that it's almost always better than label propagation.
Cool, thanks. It looks like we did actually miss a question from the lightning talks for Jeremy. Jeremy, where did you go? Are you still in the room? Oh, he left. Well, they asked if Fuser could be can be used to represent evolutionary relevant gene fusions or fusions produced by trans-splicing. And if yes, how easy would it be to be tuned to this work for other species and even across species? Oh, great. Thank you.
I can answer that question for Jeremy. I'm Dr. Wagner. I'm the PI of Jeremy's lab. Yes, Fuser can, in fact, represent trans-splicing fusions. And it uses a universal framework for representing fusion partners. And you can specify kind of the originating event for the fusion, including trans-splicing events.